Most developed countries have witnessed an unprecedented increase in the rates of obesity and metabolic disorders over the past few decades, which has spread to developing countries and become a global health problem. Alarmingly, the prevalence of metabolic syndrome in men of childbearing age is increasing worldwide, mainly due to the intake of excessive energy, resulting in male infertility and caused trouble for couples. Several studies demonstrated that gut microbiota plays a pivotal role in MET-S. The gut is ideally situated to sense an excessive energy status and is capable of digestion and absorption of nutrients via gut microbiota, further altering whole body metabolism. However, fewer issues focused on the way in which the gut-testis axis affects male infertility induced by MET-S. The new findings in our study are as follows. 1. A MET-S model induced by diets with excessive energy levels resulted in a notable elevation in the number of abnormal testicular tubules and a sharp decline in spermatogonia differentiation. 2. The vitamin A absorption was disrupted, which mainly due to the dramatically reduced abundance of bile acids induced by an imbalanced gut microbiota in the MET-S model. 3. The abnormal metabolic effects of vitamin A were transferable to the testicular cells through circulating blood, which contributed to the damaged spermatogenesis. 4. The abnormal vitamin A metabolism contributed to damaged spermatogenesis in a gut microbiota-dependent manner by using fecal microbiota transplantation. In detail, to study the influence of diets with excessive energy levels on spermatogenesis, we established an excessive energy diet fat sheep model. Compared with the normal diet fat sheep, the high-energy diet group showed clear signs of metabolic syndrome, which was accompanied by a significant reduction in the number of spermatogenic cells and a thinning of the spermatogenic epithelium. Furthermore, the epididymal spermatozoa concentration was lower, and several cell debris was observed in the MET-S model. To define which spermatogenic cell subtypes were engaged in biological pathways or functions during impaired spermatogenesis in excessive energy diet-induced MET-S sheep model, 10x genomics analysis of testicular cells was performed, and we observed that spermatogenesis was impaired mainly due to a hindrance in the development of spermatogonia differentiation. Subsequently, to address whether the arrest of SPG differentiation was closely connected with gut metabolism, we examined the untargeted metabolome profiles generated on intestinal digester samples and found that the absorption of fat-soluble vitamin A was disturbed due to reduced bile acid levels in the MET-S model. The gut microbiota plays a vital role in its host's metabolism by interacting with the host signaling pathways. Therefore, we examined bacterial 16S rDNA sequencing of intestinal digesta and revealed that Ruminococcaceae NK4A214 group was dramatically downregulated, which in turn had a profound influence on bile acid levels and further affected host vitamin A absorption in the MET-S model. In order to deeply investigate the mechanism of impaired spermatogenesis, we verified vitamin A metabolism in the serum and testis and found that vitamin A levels were significantly decreased in the MET-S model. Vitamin A can be transported into testicular cells via retinol-binding protein 4. RDH10 is essential for the production of retinoic acid, which is indispensable for SPG differentiation. Therefore, we examined the RBP4 and RDH10 levels and found that they were downregulated, resulting in the decreased expression levels of STRA8 and SYCP3 in the MET-S model. Furthermore, we performed fecal microbiota transplantation experiment to confirm that perturbative vitamin A metabolism led to impaired spermatogenesis is in a gut microbiota-dependent manner. Collectively, these findings define a starting point for linking the testicular function and regulation of gut microbiota via a host of metabolomes and the regulation of vitamin A metabolism through restoring the gut microbial ecosystem will be of value for male infertility therapy in individuals with MET-S.